Okay. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. Hello all. Welcome and thank you, Symbis people, for joining us the 54th seminar, and I call it a KAIST Day. I just resumed my seminar or presentation trips, returning temporarily back to home from Stanford University after meeting many great people discussing synthetic cells. I'll go to University of Illinois, Urbana-Champaign immediately after this seminar today, followed by my talk at University of Wisconsin at Madison as well. And I will have 7 more weeks of trip to give 17 more talks before the end of this year. Okay. It is my tremendous honor to introduce very briefly our pioneer speaker, Professor Byung-Hwan Jo. He is a KAIST endowed chair professor, visiting professor of Denmark Technical University, steering committee member of Asian Synthetic Biology Association, Young Scientists of World Economic Forum, a member of Korean Academy of Science and Technology. He is also traveling a lot, like me, currently in San Diego, rather than Korea. So probably better time zone than Korea, now midnight, I believe, right? He just got there, I think, last night, and I guess he's still in Korean time zone, of course, or in the middle. In other words, midnight, I said, or somewhere 4 a.m., I don't know. Therefore, I thank you more for your time and giving your talk for young people. In fact, you are the second pioneer from Korea. The first was Professor Sang-Hyun Lee, and he gave his amazing talk and stayed until 1 a.m. 1 year ago, and then slept probably a few hours to catch his train to Seoul in Korea. So Professor Jo, the virtual podium is all yours now. Thank you.
All right. Thank you very much for your great introduction for me. So my name is Byung-Hwan Jo from KAIST, Korea. So my department is biological sciences. So my department has many different disciplines. So I have many good peers in my department. So my lab, let me share my screen. So to prepare this great opportunity to meet everyone in synthetic biology field, I decided to share my CV rather than share scientific slides with you guys. So I just arrived at San Diego maybe 4 hours ago. So I got just a phone call from Professor Moon. So oh, wow. So anyway, so I'm now professor at KAIST Korea, and then my lab has about 25 people. Then they are studying about this synthetic biology, many different topics. So I graduated as just Professor Moon introduced. I graduated. I did my PhD at Seoul National University for enzyme engineering. So I did engineering of many different industrial enzymes. And then after graduation, then I decided to do the postdoc. And then I was searching my postdoc place in the United States. So then one of my academic brother or one of my senior suggested to go University of California, San Diego, Professor Bernard Paulson lab. So as some of you guys already knew, Professor Paulson is doing the computational modeling of metabolism in bacterial or eukaryotic cells. But I did I did not have any information about that modeling part because I did my PhD for enzyme engineering. So it was my it was very hard to decide whether to go there or not. But I thought it was I thought that modeling is very very interesting discipline because my undergraduate is chemical engineering. So that chemical, the metabolic modeling is based upon somehow that chemical engineering department discipline, I thought. So then I thought it will be really interesting, to learn the modeling, metabolic modeling of cells. Then I decided to go there. Then I spent several years to learn metabolic models and many other things. So it was very good moment to me because I published many papers with another person. So and then the place was San Diego. It's a sunny place, and then I could enjoy my life over there. So then I the change. I decided to come to Korea to join that KAIST Department of Biological Sciences, and then I started my new job at KAIST. So if I look back my career development, then I didn't thought about career itself. I truly enjoyed my life after PhD, and then before getting job at academic positions. So I thought the most important thing for young people, especially you guys, the most important thing is to enjoy your life and enjoy your research. So then an academic position or the career development will be automatically obtained in a really good shape. So that is my kind of words for you guys. So look at this. After that, I got many invitations from many different places. So including the DTU, Denmark Technical University, and even the UCSD. So I'm enjoying my life for many different roles in academic position and in industrial position and even the government positions. So so again, the most important thing in the career development is to enjoy your life for making the better career or your future. Then everything will be followed by that. So my research areas, look at this, my research interest, it's very different from my postdoc field. So I learned many things about the modeling part. And look at this, I'm doing very different researches right now. So the bacterial engineering, metabolic engineering, or the microbiome engineering, because my students in my lab, they're enjoying their life also. So they are choosing their topics for their career development as well. So I think this is my very important my discipline for making my life. So I really share my thoughts about this with you guys today, right? So this is everything for sharing my thought with you guys, right? Yeah. Thank you for the invitation to this great SIM-based activity. And then I really, really thank, for Professor Moon to make this great opportunity for everybody in the world in the field of synthetic biology. Thank you very much.
Thank you so much. That is amazing talk. And you know what? Like now you remind me of one comic book I read or enjoyed when I was very young. That's about Kung Fu. And the comic book, the teacher asked the student who would be the best Kung Fu person, and then what they are doing. And then the student basically answer like somebody who practice very hard, that might be the best Kung Fu person. And then teacher said, no, the best person is who enjoy Kung Fu the most. And then that's absolutely right. I mean, if you enjoy your life and what you are doing, you will master whatever you are doing. If you just try hard, you will be good, but not great, I believe. So that's fantastic. And also you change your field from one, enzyme engineering, to modeling, now changing another to another. And I believe many pioneer kind of changing their field from one to the other because they have too many interests in different directions. So, and also I now also realize Bernard Parsons is my academic great-grandpa.
So that's right.
You're my grandpa, academic grandpa, so somehow. But thank you so much. I mean, that's absolutely brilliant talk. Thank you.
Thank you very much.
Thank you so much. All right, okay. Now the main speaker of today with a longer introduction, Dr. Witten Schroeder, majored in both chemical engineering and mathematics at Iowa State University, another interdisciplinary kind of training. While at Iowa State, he was an undergraduate researcher. Upon graduation, he enrolled in the doctoral studies program at the University of Nebraska-Lincoln. He joined the Systems and Synthetic Biology Laboratory advised by Dr. Rajiv Saha in 2017 and graduated with his PhD in 2021. His areas of research as a graduate student include building optimization-based tools for genome-scale models of metabolism and synthetic biology. He focused on designing and modeling genetic circuits. He also involved himself in several professional development pursuits to improve himself as a faculty candidate. He was a 2017 fellow for the Science Outside the Laboratory program in Washington, D.C., and 2020 fellow of the Preparing Future Faculty program at UNL, and received a graduate minor in college STEM education. In 2021, he became a postdoctoral scholar advised by Dr. Kostas Maranas at Penn State University. Of course, he's, Kostas is one of my, best friend in academia. And as a postdoc, his research focused on kinetic models of metabolism for cyanobacteria and new numerical tools for these models, including applying systems biology to synthetic biology. Without further ado, I will now turn over speaking to him so he can share the exciting works. And thank you so much for your time, and please take it away. Thank you.
Yes. All right. Thank you, Tae-Sok, for the introduction. And thank you, Dr. Cho, for
sharing your thoughts and insights. Actually, that introduction is a little old. I wrote that last year and then my projects changed up a bit. So you'll see some of my new projects here as part of this presentation. So today I would like to talk to you about something that's not usually considered in synthetic biology, which is systems biology and computational approaches there too. Considering that, why is the clicking not working? Oh, there we go. So I just wanted to highlight some of my work. So I've published 8 different articles, 7 of which are first author, several different types, including research, protocol, and review. And I think this is very indicative of my approach to science, as I hope you'll see throughout this presentation, because I want people to use my work in their own work and carry it further. So I'll start by giving a brief introduction as to what systems biology is. And since I realize I'm talking to mostly synthetic biologists, just so that we're all on the same page, then what I'll do is talk about some of the different projects that I've been involved with and the biology thereof, because my PhD thesis was actually in tools for genome scale models. But considering we have a synthetic biology audience, you might not be interested in the heavy math. So hopefully we'll talk a bit more about the biology to keep you all interested. So first, I'll talk about defensive pigments in a fungi and the cost thereof and the synthesis thereof. Then I'll talk about how I built a life cycle plant model for Arabidopsis. Then we'll talk about how we used metabolic modeling to study root reprogramming under nitrogen starvation. Then we'll talk about sort of my 4th project here, which was possibly the most interesting to synthetic biologists, which is the optimizing of genetic circuits using modeling tools. And then finally, we'll talk about some of my work I've done as a postdoc, which is reevaluating atypical glycolysis. So we'll start with an introduction so we're all on the same page. So systems biology is an interdisciplinary intersection of mathematics, biology, and chemistry. So from chemistry, we get sort of our basic chemistry ideas like kinetics and stoichiometry. From biology, we get ideas like the central dogma informing what capabilities that organism has. And from mathematics, we get our framework for describing our model. So this is just sort of the basic chemical formula mathematical formula of our models. Often we'll assume our models are operating at pseudo steady state, and then this is called a stoichiometric model or an SM. And if it contains all genome supported reactions, we call it a genome scale model, often called the GSM or GEM. Building these models is a very time and labor intensive process that is iterative. So we try to bring together as many sources of data as we can in order to create draft models. We'll refine those based on various analysis techniques. Then we'll have our model, and then we'll return to using other analysis techniques and tools in order to see what we can learn from them. So the total time for model building is anywhere from 6 months to several years. And according to sort of the standard protocol, there's about 96 steps in model reconstruction. However, once you have a model, then you can continue to answer lots of different questions very quickly once you have a model that you have confidence in. So for instance, 2 of the main ways these models get used is for gene knockout or change in gene expression in order to create some desired bioproducts. Another way we can use these models is to discover novel biofunctions by noticing the incongruities between our models and what we know happens in the living system. So ideally, model building can save us time, labor, supplies, and grant money in the long run. And I think this supplies is a better argument now since COVID, considering we've had all these supply chain disruptions, including for lab materials. So we'll talk about sort of my first application of stoichiometric modeling. So the melanized fungus, Exophylla dermatitidis, has 2 defensive pigments called melanins and carotenoids. And this protects it from a lot of extreme conditions. So it's actually quite an interesting organism. It's a eukaryote, but with a very small genome. an order of magnitude less than other model systems, and its proteome is also an order of magnitude less. We also see that the key enzyme in melanin production, the tyrosinase, is very well conserved amongst many different species. So our research question in particular was what is the cost of these pigments to our organism? And then can we use our organism as a model melanin producing system, given its highly conserved tyrosinase and rather small genome and proteome? So we addressed these questions using stoichiometric modeling. However, we ran into a problem very quickly in this study, which is that Exophylla dermatitidis is not very well studied. So to this effect, we actually created a whole workflow to try to fill in gaps in our knowledge using information from a related set of organisms, the Aspergillus genus. And to systematically apply this, we created the OptFill gap filling tool. So this was actually my first publication, and this addressed a couple of issues that we had, and I'll discuss it briefly on the next slide. But in total, this resulted in us creating a new genome scale model called IEDE 2091, which has about 1,700 reactions, 1,900 metabolites, and 2,100 genes. So it covers a very large percentage of the proteome, at least for a stoichiometric model. So to be able to do this, we had to create our OptFill tool. So our OptFill tool is a two-part algorithm for thermodynamically infeasible cycle-free gap filling. So this first step here, this TFP, basically finds all the infeasible cycles that could occur between your model and your database. And then we have this second step, which is the connecting problem. And this first step informs the second step by saying these cycles are not allowed. And we can also quickly modify this to identify our inherent infeasible cycles to much more quickly curate our models. Our second step is our connecting problems, which basically minimizes the number of new functionalities added to get the maximum number of metabolic gaps filled. So that's why we call this a conservative and a holistic approach, because we're minimizing and doing it on a whole model basis. So this will hopefully also minimize our dependence on who the manual curator is. So our results aren't dependent on who the researcher is is more dependent on the biology. So when we when we reconstructed this model, we first applied shadow price analysis to it in order to look at the cost of these defensive pigments. So shadow price analysis is a technique most commonly used actually in economics, and it basically tells you the cost of something for which there is no market, or the cost of producing something, one more unit of something. So we use sort of that second definition in order to study the cost of melanins and carotenoids in Exophylla dermatitidis. So we find that the cost of both is rather high. So its cost is actually greater than the growth rate most of the time. And we find that carotenoids, pretty much irrespective of which carotenoid we're looking at, is more expensive than our melanins. And this struck us as rather odd because, as we can see from this figure, the melanins are deposited in the cell wall, whereas the carotenoids are deposited in the cell membrane. And there's no function that we know of that carotenoids can fill that melanins cannot also fill. So this led us to our first hypothesis about the biology of the system, which is that carotenoids have an as yet undiscovered role in Exophylla. And our second hypothesis comes from the fact that we see that the different molecules of each category change costs depending on what we fed the organism to grow. So our second hypothesis is that our species undergoes some kind of cost minimization of the total defensive pigment profile, depending on what resources are available. So I wanted to highlight that some overlap exists between Exophylla and human metabolism for melanin. Specifically, if we look at the two types of melanins that humans have, eumelanin and pheomelanin, the key difference in the branching between these two pathways is the presence or absence of cysteine, and then it's spontaneous reactions from there on out. So we believe that if we grow Exophyella in a cysteine-rich culture, that we could induce pheomelanin synthesis and then have both melanin classes that humans have. There's two other melanin pathways that Exophyella has, the pheomelanin and DHN melanin, but those are unique to Exophyella with respect to humans. We also looked at the active sites of the key enzyme in tyrosinase, the key enzyme in melanin synthesis, which is tyrosinase. And we find that these active sites are quite well conserved. So the pink indicates sort of the highest level of conservation. The blue indicates moderate conservation, and the gray represents non-conservation. And the top set of enzymes are humans, and the bottom sets of enzymes are Exophyella. And we can see that many of the key residues highlighted up here are well conserved. And we even have some conservations of residues that are whose mutation is indicative of the most common type of albinism, which is called oculocutaneous albinism A1. So overall, we think that we can use Exophyella as a model melanin producing system, but we'll need a lot more in vivo verification before we get there. So we have 3 publications involved in this work. So all 3 of them involve OptFill, and 2 of them describe our Exophyella model and how we use this for our shadow price analysis. So if you're interested in my work, please check out my papers and protocols. So I've been rather system agnostic throughout my time as a PhD student, and at this time we actually transitioned to doing grant work on plants. So I'll talk about some of my plant work next. So we're part of the Center for Root and Rhizobiome Innovation, or CERI, at the University of Nebraska-Lincoln. And this center is basically a multi-pronged for a multi-lab approach to try to address plant health and yield through manipulation of the root system via synthetic and systems biology, and to try to better understand what's going on here. And this is because, and for those of you not from the Midwest, corn is a big staple of Midwestern agriculture and is often used for food, biofuel, export, and animal feed. So improving corn yield and corn health could have major economic implications throughout at least the Midwestern and certainly most of the United States. So our goal as modelers was to try to better understand root biology and how the rhizobiome and root interact, as well as how they interact with the rest of the plants. So our first goal, because this was a new center and they were still trying to put together all the data that we needed to build our model, was to ask, how can we model plant scale properties over a long period of time, such as a growing season? Now, since we didn't have all the maize data that we were hoping to have, we instead decided to apply our modeling techniques first to the model plant Arabidopsis thaliana, so that we can then port those techniques over to maize at a later time. So what we did was we broke down Arabidopsis thaliana into basically 4 representative tissues, each with a unique function based on their role in their native systems. So for instance, the leaf model was modeled for gas exchange and photosynthesis. The root model was modeled for respiration, transport, and nitrogen reduction. The seed model was modeled for storage, as well as to model the sink that comes along with reproduction. And the stem model was modeled for transport, respiration, growth, and as a way to logically link the other tissues. And I should point out at this point that this is a core model. These models are very small and not genome scale, but this is for a test of creating whole plant models. So currently, if you want to model across a long period of time, or at least at the time I did this research, you had to use a dynamic flux balance analysis approach, or DFBA approach. At the time, there was 2 approaches. The first was the static optimization, or SOA approach. It's also in a step-by-step manner. It's easy to implement and utilizes linear programming. However, it utilizes first-order Taylor series approximation. So this limits our solution accuracy and stability over time. The other approach was the dynamic optimization approach, or DOA. This solves all time points simultaneously. It's accurate and the solution is stable. However, it requires nonlinear programming, and this makes things difficult to implement and scale up. So we sort of split the difference with our new approach to DFBA, which is called the Optimization and Explicit Runge-Kutta-based approach, or ORCA. It improves on the SOA because, like the SOA, it solves in a step-by-step manner. It's easy to build and utilizes linear programming. However, unlike the SOA, we can choose different Runge-Kutta methods to implement for our ODE solver, and this gives us a tunable accuracy and stability for our models. So in this particular study, I used Hume's third order rule, and as well as if I had to do any integration, I used a trapezoid rule approach. And for derivatives, I use a backwards finite difference approach. So the ORCA to DFBA allowed us to create a 7-stage life cycle Arabidopsis model. So the stages you can see in the figure here. And this is a significant improvement over what was enabled by the previous methods, because now we're able to model from germination to 61 days. We can take time steps of 1 hour. Our step error is equivalent or better to what we see in previous DFBA approaches. We're able to model 7 growth stages, and we're able to model 4 tissues as opposed to 2. So this gives us a modeling framework for a whole life cycle model of a plant, which we're hoping to port to maize in future. We also looked at the plant scale model behavior and found that it matches experimental observations. We did this by looking at yields of different tissues, specifically the stem, the seed, and the leaf. And we found that our yields were well within 1 standard deviation of experimental measurements. So we were quite happy about that. We can also use the particular model to study flow of sulfur metabolism in water across the model at all time points. So for instance, for sulfur metabolism, we saw just about what we expected to see. So the root as a source, sulfur assimilation in the leaf, and a sulfur sink in the seed. So now that we have our framework, we started moving toward building the individual models of the maize plant. So we started by building a maize root model. And what we wanted to study with maize roots was how it changes under nitrogen stress. So we can see here in this image that if we provide full nutrition and nitrogen starvation to maize root, we see that under nitrogen starvation, maize root grows much faster and much longer and has increased mass compared to the shoot. So we're hoping to recapture these phenomena in our model to say that we have a good model. So the modeling efforts was actually a collaboration between my postdoc lab, the Maranis group, and my PhD lab, the Saha group. And we actually got data specific for building these models from the HERO group at INRAE. So they collected transcriptomic data for us. So the ultimate research question that we wanted to answer here was, under nutrient stress conditions, what metabolic reprogramming drives our phenotypic changes? So briefly about how our transcriptomic data was gathered. So the group in France, the HERO group, hydroponically grew maize. They then did gene expression profiling under nitrogen deficient and sufficient conditions using a cDNA microarray. They then identified key metabolites that might be indicative of the metabolic reprogramming from their knowledge of literature, and extracted and analyzed metabolome information for those particular metabolites. We then took the information that they gave us, particularly the transcriptomic data, and used that to influence how our model behaves using the eFlux algorithm, which basically is a valve-based approach to influencing how much flux is allowed through each reaction in a pathway. So based on how the gene is upregulated or downregulated, this allows us to turn valves, which then changes how much we allow through different metabolic pathways. So this allows us to partially model our regulatory changes as well as our metabolic changes. For analyzing how concentration changed, during our starvation conditions, we used a proxy called flux sum analysis, which is basically looking at the average of the total flux through that particular metabolite. And we call this metabolic pool size. So our transcriptomic data was integrated into our two models, our nitrogen sufficient and nitrogen deficient models. And we saw that for many of these, we had a strict increase in our metabolic pool size, which is indicative of higher biomass growth. So this was good news for us. But we also saw that our lipids had highly variable flux sum ranges. So we dug deeper into our data and into our model to see where this came from. So we ultimately saw that phosphatidylcholines and phosphatidylglycols were the culprits here because they had very high flux ranges, and these flux ranges were not well linked to growth rates. So it seems that they're able to store carbons or energy in these particular molecules without necessarily contributing more to biomass production. This particular work is published in the Journal of Experimental Botany, if you're interested. We also looked at how nitrogen starvation changed our flux ranges through key pathways in metabolism. So for instance, we saw that there was increased metabolism through various pathways that help fix nitrogen, such as the sphingolipid metabolism and the benzoazonoid metabolism. These metabolisms can attract nitrogen fixing bacteria in order to promote nitrogen fixation. So we were excited to see that. We also saw that very carbon heavy metabolisms had increased flux rate. This is probably indicative of storing more energy for hopefully a better time when nitrogen is available. We saw increased metabolism through ATP producing pathways as well in order to sustain our increased growth. And we saw increased production in the root, at least, of amino acids so that we can build more root structures and have larger roots. So we were very pleased with these results, and we think that the trends match the trends that we're seeing in the literature and in the biology. So this is another project that I did for the CIRI group, or the Center for Root and Rhizobiome Innovation, which is as part of building our models, we also want to have some way of creating strong control of metabolism and the interactions within the root and between the root and the rhizobiome and the root and the plant. So here's where we created genetic circuits. So sort of a brief introduction to genetic circuits. I mean, I know I'm preaching to the choir here, but this will have relevance to the talk in a sec, I promise. So genetic circuits were first introduced around 2000 with the advent of the repressilator and target and toggle circuits. And circuits are, and pretty much still are, dependent on rational and intuitive design. So you need an expert in genetic circuits or an expert in synthetic biology to be able to use these synthetic biology tools. So as a computational biologist, I want to take a step back and say, okay, can we design these kind of tools without expert knowledge via some kind of tool or optimization approach? So that was our research question here. And this could lead to a couple of different advantages. For instance, using optimization, we can often get non-intuitive yet highly effective designs. And as I said before, this could reduce the expert knowledge needed in order to create this most basic of synthetic biology tools. So we did need to have an application for this. So our thoughts was phytoremediation. So what is phytoremediation? Phytoremediation is the use of plants in order to somehow remove contaminants from soil. The advantages are it's eco-friendly, very cost-effective, and there's several different strategies you can use, such as shown in the figure here. And our particular application is the phytoremediation of heavy metals using corn or ZMAs. And this is because ZMAs is a natural accumulator of heavy metals anyways. So we were hoping to build on the natural competencies of maize for this particular root application. So the scope of the current work is actually going to be a couple of steps away from our long-term goal of remediation. Namely, we're going to be designing genetic circuit sensors in Arabidopsis thaliana that we hope to be able to port to maize so that we can start building our phytoremediation strategies a few years in the future. So we're creating our sensors using the Eugene SID and Eugene SID-M workflow, which I'll briefly describe here. So this workflow is very much on the design part of the design build test cycle. As I'd like to indicate here. So first we sort of conceptualize how we want our root system to respond. We then create a library of parts, which include promoters, transcripts, and terminators, which we think might be able to accomplish this particular response that we want. Then our first tool, the Eukaryotic Genetic Circuit Design Tool, or Eugene SID, as I've been calling it, puts together triads of promoters, transcripts, and terminators, which when taken together will give us that desired behavior, at least in a static way. We then investigate the dynamics of that behavior using the Eukaryotic Genetic Circuit Modeling, or Eugene SIM tool. So this helps us screen our designs to make sure that there's no problematic dynamic behaviors that we aren't seeing in our static solving. We'll then repeat this until we get a desired number of solutions or until we reach some type of other termination criteria. And then we'll screen a large set of design solutions. So one advantage of having numerical methods for designing is we can create dozens, hundreds, thousands of solutions against which we can screen. And then finally, we can select a set of designs to build in the system. So Eugene SID-M, this is basically the workflow. So first we have a conceptualization where we have a logic table input to say how we want our system to react to certain input signals. We'll then feed that to our Eugene SID tool, and that'll create sets of triads which will have our desired behavior, including producing, in this case, producing a green fluorescent protein signal. And then these triads are passed to the Eugene SID-M tool in order to see how the dynamic circuit behaves. So for instance, here we see an OR circuit to cadmium and copper, and we see correct dynamic behavior. Here's another example where we have a non-implication circuit between copper and zinc. And we see that a buffer got added here to give it the correct dynamic behavior, but a very long startup period. And we can also see here that, for instance, in a cadmium zinc not and circuit, we get correct or a design from Eugene SID, but incorrect dynamic behavior when we model it using Eugene SIM. So this just allows us to screen out potentially incorrect designs. And this incorrect design actually came from too leaky of a promoter, allowing too high of a buildup of this particular regulator, which turned off green fluorescent protein when it shouldn't have. So we are also considering some of the biggest problems for genetic circuit design, which is promoter leakiness in our tool. So we hope that people will find this quite useful. We also use the standalone Eugene SID-M tool to model a repressilator circuit. Here we have two fluorescent proteins, a yellow fluorescent protein and a green fluorescent protein. And we can see that we're able to model both startup and sinusoidal behavior for transcript and enzyme levels to describe how the circuit is behaving. And we're also glad to see the shape of the circuit behavior that we do, because this is sort of the shape of the circuit behavior that we see in vivo. So even though we're using mixed integer linear programming, we're able to get some nonlinear dynamic behavior out of because of how we set up our tools and how we set up enzyme degradation. So the Eugene SID and Eugene SID-M tools are published along with a protocol for its use if you guys are interested in potentially applying these to your own research. And then finally, we're going to talk briefly about some of the work that I've been doing as a postdoc in the Maraniss lab, which is reevaluating atypical glycolysis. So when I joined the Maraniss lab, I joined the Center for Bioenergy Innovation. It's one of the 4 bioenergy centers from the Department of Energy. And the goal of this bioenergy center is to essentially take process advantaged feedstocks, lignocellulosic feedstocks, and convert them to sustainable aviation fuels via a multi-step process. So this is done by consolidated bioprocessing with co-treatment of Clostridium thermocelum, and then catalytically upgrading those products into sustainable aviation fuel, which includes cycloalkanes, branched alkanes, and aromatics. So our role in the center is to model Clostridium thermocelum and its bioprocessing of the lignocellulosic biomass. Now, Clostridium thermocelum is a really good degrader of lignocellulosic biomass, but it also has a lot of atypicalities, which make it kind of difficult to work with at times. So we can see here that shown in the blue arrows are the atypical glycolytic steps, and in the blue box is our target metabolite, or ethanol, that we're hoping to produce a lot of. So to be able to produce a lot of ethanol, we need to be able to understand the atypicalities of glycolysis. So particularly interesting is that guanosine triphosphates often replace adenosine triphosphates, and sometimes even pyrophosphate replaces adenosine phosphates in glycolysis. So there have been previous attempts to try to identify where this pyrophosphate source is coming from, which powers glycolysis. So the most recent one was in 2021, where they knocked out these 3 steps highlighted in orange, which they thought were going to be the primary contributors of pyrophosphate to the system. And they found that this decreased biomass yield only about 28%. So their conclusion was there's some other way pyrophosphate is being produced. So my research question that I'm addressing with stoichiometric modeling is, using this latest set of data, can we identify the primary pyrophosphate source through modeling? So unfortunately, I don't have a happy ending here yet. So far, our results to identify the primary pyrophosphate source have been unsuccessful. So we tried to do this by reconstructing a genome scale model of this organism focused on phosphate metabolism with extensive curation and validation against 10 different strains. We then applied the OptStoich and MinFlux algorithms to try to identify where our pyrophosphate is coming from. So OptStoich is a way of trying to maximize our the thermodynamically favorable yield of pyrophosphate using this canonical pyrophosphate synthesis equation. And MinFlux uses the model to try to identify what pathways can produce this particular stoichiometry. So from our MinFlux algorithm, we found only 3 solutions to produce this canonical, as favorable as we can, yield of pyrophosphate. That's glycogen cycling, the membrane-bound pyrophosphatase, and the malate shunt. But we know from our in vivo experiment published in 2021 that these can't be our main pyrophosphate sources. So we needed to explore other avenues of pyrophosphate synthesis. So our other attempt was to investigate directly through the model to try to find other pyrophosphate sources. So we found that upwards of 67 reactions were able to produce pyrophosphate at some level, but only 18 of those were able to produce high levels of pyrophosphate, enough to sustain glycolysis need. And they break down into 2 schemes. So the first scheme is a nucleotide triphosphate pyrophosphohydrolase, which basically turns a nucleoside triphosphate into a nucleoside monophosphate while producing pyrophosphate. And the other scheme scheme 2 basically does the same thing, but through a multiple enzyme cycle. However, these mean that the cost of pyrophosphate is 2 ATP, which means that the pyrophosphate substitution of ATP would be energetically unfavorable throughout glycolysis. So these are likely not our main sources of pyrophosphate. So this leads us to 3 different hypotheses for why our model hasn't currently been able to find our main pyrophosphate source. So first, we hypothesize that there's some currently unknown or unannotated function that we need to add to our model to resolve this incongruity. Second, we hypothesize that pyrophosphate generation might be by a mechanism outside the scope of this model. Or we hypothesize the less favorable pyrophosphate yield is acceptable by high reliance on the malate shunt. And this is why we're seeing a decrease in biomass yield in vivo. So the question is, aside from these hypotheses, what can we take away from this modeling effort? So even when the model fails, we can still use our expert knowledge applied to the model, I think, to still learn something fundamental about the biology. So in this case, we learned about the stringent response to starvation. So the stringent response in Clostridium thermocelum is accomplished by this bifunctional bistate REL-A-SPO-T enzyme. And when C-therm is starving, it'll use GDP and GTP or ATP and fuse those into a PPGPP alarm, which reduces growth and also depletes ATP equivalents within the cell very quickly. On the other hand, when it's not starved, this SPO-T enzyme is active and it hydrolyzes this alarmone into GDP and pyrophosphates. And we noticed some really interesting characteristics of this system while we were building and analyzing our model. First, that it has very high gene expression, so anywhere between the 83rd and 90th percentile of gene expression, and this correlates well with growth. We also saw a high concentration of this alarmone in the related organism B. subtilis, and this was up to 90% of unstressed GTP concentration. And then finally, we found that this alarmone hydrolysis actually produces our glycolytic cofactors. So pyrophosphate and GDP or GTP are produced. So this leads us from the knowledge we gained from model building to the hypothesis that atypical glycolysis cofactors allow for quicker recovery from the stringent response to starvation. So this is sort of an exciting case study because the model in its for lack of a better word, intended purposes has not succeeded, but we found some other interesting insights about the biology of the system that we're excited to publish shortly on. And with that, I would like to thank, well, a lot of people actually. So my mentors, my research mentors, including Dr. Shanks at Iowa State, Dr. Saha at Nebraska, and Dr. Moranis at Penn State now. I'd also like to thank collaborators with whom I've worked on these projects with, including Niaz Chowdhury at Nebraska and Mohammed Islam at UNL, as well as thank in vivo collaborators who have given us data to work with for our modeling efforts, including Bertrand Hurel at INRAE in France, and Dan Olson, who is at Dartmouth. I would like to thank our funding sources, which have been, or my funding sources for these projects, which have been the Center for Root and Rhizobiome Innovation and the Center for Bioenergy Innovation. And then finally, I would like to thank the many mentees I've had throughout my research experience, including graduate students, undergraduate students, and high school students, all who have contributed in some part to the research presented here. And with that, I would like to thank you all for your time. And if you have any questions, well, now would be the time to ask it. Thank you.
Oh, amazing, amazing talk. Very complex, system you deliver very, nicely. Like many people understand, what you are talking. So I believe Professor Joe has lots of questions. If you want me to, ask for you, I could do that, but I probably prefer you just speaking, asking the question.
All right. So, your talk remind me many, previous works at the UCSD, but currently my lab is doing very similar topics by many graduate students. I think it's really great. And then, because you are doing some metabolic modeling work for plant and especially plant root, then I have some questions, probably one or two questions for you. So, because, in my lab, I'm really, the main question that I have is the interaction between plant root and nematode. That microbiome is a nematode, because that there are tons of nematode in the soil, and many of them are really good for plant growth, but some of nematodes are really harmful for plant growth. For example, the coffee tree. So in many underdeveloped countries, they are growing the coffee trees for their life, and then they are making money from their coffees. But that nematode is really serious problem for coffee tree growth. But as we know, there are not many chemicals to specific, inhibition for the harmful nematode only. Because when we treat the chemicals, then every nematode will die. But some of nematodes are really good for, plant growth. So here, the question. So I think if we look for the nematode life cycle, then the plant root metabolism is really important for nematode infection, because not every time the nematode infected plant growth. So do you think, can we predict the, some metabolic state of roots? And can we design some, I don't know, this is my just thinking. So can we design the plant root metabolism to protect the nematode attack, nematode infection, right? This is my really great question. I mean, this is really important for, agricultural industries, yeah. So this is my question. Yeah.
Yeah, we very much could do that, and I think that's part of the reason I had some of the models I did, right? So that's part of the reason I made the root model, is so that we can then add on other models of other organisms in the ecosystem and take a look at those. There's actually was some quite vibrant discussion in the center while I was still there, because that was my PhD lab. I haven't kept up on what they're doing recently, but there was some quite vibrant discussion that possibly corn roots actually export some metabolites in order to promote the growth of certain targets, microbes that are very good for its health. So think of it like an investment in the bank, for instance, and getting that interest back from the microbes. And we didn't look at nematodes yet, because we were at that point, we still didn't know all the members of the microbiome yet, so we hadn't moved on to multicellular organisms yet. But I'm sure nematodes play just a huge role, because I mean, I'm sure for instance, they're predatory, and they can change the abundances of microbes in the rhizobiome, and they probably serve lots of different functions. But it's not something I have looked into yet, but it's definitely something to keep in mind. Yeah, and I'm glad you actually mentioned coffee, because that was one of my pet projects as a grad student, was to try to get funding for a USDA funding to study coffee, make a metabolic model of coffee to see if we can make it more robust to climate change, because coffee is a very fickle plant, and climate change could really disrupt our supply of it. So coffee is one thing I'm quite interested in modeling for plants in future. Yeah,
fantastic. Yeah, because one of the reasons for that is climate change, of course. Due to the climate change, many countries struggling with that some unexpected problem with for example, the nematode infection. So so that's why I ask what do you think about this metabolic design for. Of course, we can use the metabolic design for increasing the productivity and production of new chemicals, but at the same time, we can think about the metabolic design for protect unexpected infection by other organisms. So that's very natural thing. So yeah, that's why, because synthetic one of main goals of synthetic biology is a grand challenge for the for the climate change problem. So that's why I'm asking you about this.
Yeah. And I think that was part of the point of developing the Eugene SID and Eugene SIM tools, so that we could have not just a single metabolic state, but we could have inducible metabolic states to various stimuli. That work was really in the early stages where we were just building a design tool. But I'm hoping to see that work sort of take root and blossom so that we can start making some really interesting applications, because as I mentioned, corn root's a natural accumulator of heavy metal toxins. So that'd be really cool if we could design a corn plant that turns on phytoremediation only for as long as we need it, turn it off when it's done, and still maintain its yield. So that would be a really cool project.
Fantastic. Thank you very much.
Yeah. Thank you for giving me this opportunity to speak, and thank you for your kind introduction.
So Dr. Joe, do you have any additional question? You type multiple, but I believe you cover all, or you have additional question?
No, I don't have any further questions.
Okay,
this was
wonderful.
my main question. Yeah.
Okay, thank you. So I guess now we basically the 11:03. So I have question, but I ask after this formal session, and I'm basically conclude. Okay, so thank you all for joining and staying today. We'll meet again next week on October 13th, Thursday, the same time, the same Zoom link. We'll have Dr. Susan Jones, the chief editor of Nature Microbiology, and Dr. Jinjin Diao, my postdoctoral fellow. And as usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists. And thanks. I stop recording. Just give me 1 second. Okay.